introducing the aims of discovery medicine kind of as a whole to better frame the aims of the study that I've tried to do here. So, this is to identify new molecular techniques or entities that may be of value in treating diseases that present with unmet medical needs. And so, the new molecular entity that myself and my lab work on are human amnion epithelial cells and, of course, the extracellular vesicles that they produce, and I guess that's why I'm speaking here today. And so we look at it in a variety of different ways. We ask, what do they do? So looking at their biological activity via different functional assays. We ask how we make it, so addressing the limitations associated with scalability, storage, and transport, and asking, of course, how we can use it. So this is kind of where I fit into the puzzle of my lab, investigating the therapeutic applicability that these new molecular entities have in different disease contexts. And so the disease that I'm focusing on currently is called chorioamnionitis. So this is defined as the bacterial colonization of the fetal membranes and the fluid that surrounds a fetus during development. And this is important to study because this exposure to bacteria we know causes brain inflammation that results in brain injury. It exposes the fetus directly to bacteria, and this induces a pro-inflammatory response, and can also increase the risk of preterm birth, be it spontaneous rupture of the membranes or an elective seizure to remove the fetus from its adverse uterine environment. And combined, we know this results in injury that leads to lifelong debilitating disease. And so when you look at the brain, you can see direct changes to the structure of the brain, indicative of the injury that's occurred. So if you take sections of the brain from lambs that have been exposed to LPS, or lipopolysaccharide, this is a component of gram-negative bacteria that's commonly used in preclinical models to simulate or mimic the effects of chorioamnionitis in preterm clinical models. So if you look at the white matter from these brains, following an acid fusion stain, you can see here in comparison between healthy and inflamed brains, there's an increase in neuronal loss indicative of tissue damage compared there with panel B to panel A, indicated by the black arrows there. You can see an increase in macrophage infiltration, so there's big angry black spots there in panel D compared to panel E, and that's indicative of an increase in inflammation. And an increase in apoptotic bodies, they're seen in panel F with those black arrows compared to panel E, indicative of an increase in cell death. And this is important because these indices are associated with an increased risk of developing cerebral palsy, an incurable motor disease. So there has been some evidence from our group showing that these amnion epithelial cells have benefit in the context of chorioamnionitis. So in preclinical models, we have shown that administration does result in a reduction in the indices of brain inflammation. There's an increase in oligodendrocytes and myelin basic proteins. So these are the proteins that surround an axon and facilitate signal transduction through neurons, as well as a reduction in apoptosis and astrogliosis, so a reduction in cell death and inflammation. So, as we've heard over the past few days, EVs are produced by all cells, including the cells that we study in the lab. They contain the cargo of the parent cells, and relative to whole cells, from a therapeutic angle, they're easy to store, handle, and transport. And so, I'm looking at whether or not they retain the similar therapeutic properties as these parent cells. Some exciting preliminary data from our group have shown in an adult model of brain injury that EV therapy reduces indices of astrocytosis, so suggesting that there is some anti-inflammatory property, as well as an increase in myelin basic proteins, suggesting that these EVs have retained some of that reparative or regenerative properties that we've seen. So I wanted to touch quickly on why it's important to investigate fetal safety. So we might be familiar with the thalidomide story from about the 1950s, 1960s, but I wanted to tell a story about something that happened actually quite recently. So in early 2017, there was a large multi-center, multinational clinical trial investigating the therapeutic efficacy of sildenafil, more commonly known as Viagra, for fetal growth restriction. So briefly, this is when the placenta fails to deliver adequate nutrients and oxygen to a developing fetus, resulting in a stunting of fetal growth and development. And so the idea is that sildenafil increases blood flow, and so maternal administration would increase blood flow within the placenta, increase nutrient oxygen delivery, and rescue some of that fetal growth. However, this trial was unexpectedly halted early following adverse effects, which included an unexpected increase in fetal death. So to find out what was going on here, we replicated the parameters of this clinical trial in a preclinical model in sheep, inducing growth restriction and delivering maternal sildenafil. And what we found that it actually impaired the adaptations that the fetus normally mounts to survive the adverse intrauterine conditions that are associated with growth restriction. So there was a decrease in blood flow to the brain, so a decrease in carotid blood flow, as well as an increase in blood flow to the periphery. And so we think that the change in these adaptations might have contributed to what was seen clinically, highlighting why it's important to look at the effects immediate to a fetus, which have completely different physiological system compared to adults, in therapies that already have a very well-defined safety profile. I think one of the problems from this study is it went straight from small animal models into the clinic. However, when looking at first-in-human trials, we actually focus on physiological signals in response to novel therapies, and I'll touch upon this a little later on. So our group addresses this limitation by using sheep. We have established models of perinatal injury with known human equivalent developmental milestones, so we can translate the results that we see in our model pretty well to the clinic. We can perform in-depth physiological analysis following exposure to injury, insult, as well as novel therapies, and we can deliver clinically comparable doses because of the size of our model. And in a little bit special to the clinic, we can perform pretty intensive long-term follow-up studies looking at very sophisticated ways to measure and look at indices of injury as well as treatment. So for this study, we performed fetal surgery. That's myself there, my PhD student. We implanted arterial and venous catheters into a fetal sheep at 100 days of gestation. And this enabled us a window into the fetus to collect blood samples, as well as monitor heart rate and blood pressure over a period of gestation. This also enabled us to deliver LPS to mimic chorioamnionitis, and as well as deliver EVs to test our therapy. We then return the fetus into the uterus and exteriorize all these catheters out the flank of the U. We implant a maternal jugular vein catheter to administer post-surgical antibiotics, and this setup is then connected into a data acquisition system and software so that we can collect all these intrauterine physiological signals in real time over about 2 weeks. So this here is an example of what's collected clinically. This study was done in preterm babies that were at risk of developing chronic lung disease following the administration of our cell therapy, so the amniotic epithelial cells. So you've got blood pressure here in blue, you've got oxygen saturation in black, and heart rate in red. And you can see here in that first infant that there was a transient cardiorespiratory compromise. And so this is consistent with what's seen in pulmonary embolism. And so following a change in administration procedure, we don't see that for the rest of the babies, and all these babies actually did very well. So again, this just touches on how important it is to look at the physiological response to a novel therapy, especially in a fetus or a preterm which has a very sensitive and different system to an adult. So again, mimicking what we do in the clinic, so these are results from our study. I really like looking at heart rate and blood pressure because it's an immediate indice of physiological stress. So if you held your breath, or if you go for a run, heart rate increases, blood pressure increases, starts to fluctuate. What I think is interesting from our studies, we see that in response to our LPS insult, heart rate and blood pressure between control in black, LPS in red, and LPS plus EV treated in blue are pretty similar. But as time goes on, the blood pressure and heart rate in those that are not treated with EVs start to respond. We see an increase in fluctuation and variation in this group, indicative that the inflammatory insult that we've imposed upon them is causing some physiological response. What I think is interesting is that those that are treated with EVs remain close to baseline and close to control, which is indicative that our EVs might have some effect in dampening that immune response or inflammatory response that these fetuses are experiencing. We look at partial pressure of oxygen and carbon dioxide as well. So this is a concentration of these gases within the blood, and these are indices of metabolic stress. In response to LPS in our EV treatment, we see no changes between our groups. A bit of a fluctuation there during the LPS period, but importantly, no changes within the EV group, suggesting that our EV therapy isn't affecting metabolic demand within the fetuses. Oxygen saturation is another marker that fluctuates pretty quickly in response to stress, and lactate is a stress marker. So you can see in the lactate, before the EV treatment, both the blue and the red animals have a bit of a fluctuation, indicative that there is some increase in stress that they are experiencing. And then importantly, during the EV treatment, there is no change in lactate, so it's not causing an insult or an extra stress to the fetus. What's also cool about our studies is that we can look at the effects on lots of different organ systems. So I'm still analyzing this data, it's not published yet, but I wanted to give a little bit of an insight into some of the cool therapeutic things that we are seeing from our studies. So following an IBAR1 stain, which stands for microglia, these are the resident immune cells within the central nervous system, we do see this increase following our LPS or inflammatory insult. And when given our therapy, we see a phenotype that looks much more like control. We also look at the lungs. So this is just a H&E stain, a simple stain taken from the lungs of these lambs. We can measure alveolar size, density, wall thickness, bleeding, lots of different indices that suggest towards the development of bronchopulmonary dysplasia or chronic lung disease. In the setting of injury and inflammation, we see an increase in wall thickness and a reduction in the number of alveoli, indicative that this might result in poor respiratory function at birth. And then when treated with EVs, we do see a phenotype again that looks much more like control. So in conclusion, our EV therapy doesn't alter the fetal physiology compared to control animals, and this does suggest that exposure to EVs in themselves is not harmful. And while still ongoing, the histological and molecular analysis, it does suggest that there might be some improvement in markers of brain and lung injury following LPS exposure. So I'd like to thank my lab group for making all this work possible, and everyone who stayed around to have a listen today.
And it's time for question and answer. Please come to the microphone if you have some questions. If no, I want to ask a question. Did you try to do the clinical trials for using the AEC derived images for the fetal?
Not yet. No, that would be the goal after all of these studies, to do a first in human clinical trial. But I think the results of this study support that it should be safe moving forward.
Yeah.
Thank you so much for a great work. I was wondering if you do dose response and see if with different doses of EVs you've seen, I don't know, increase in the side effects or increase in the effect that you see on brain.
At the moment, we haven't done that yet. This was the first study to just test exposure. These will be the follow-up studies that we're planning to do. Of course, and I guess it's a big limitation of the EV field in general, generating the volumes needed to treat something that's 80 kilos versus something that's 8 grams has been a bit of a challenge within our group, and that's what we're working on to get those dose studies going.
If there's no more question, I have one more. So what about any other source of EVs other than the amniotic ones, like the mesenchymal stem cell ones, or any source of EVs have the similar effect too?
So in the sheep, we haven't studied that just yet. We have done some mice studies looking at the therapeutic comparison between MSCs and HAC EVs, and that was presented a couple of days ago. Okay.
Thank you.
No worries.
Any other questions? If there are no questions, let's thank again for asking me. Thank you. Our next speaker is Dr. Chong He from Jiangsu Second Normal University. Her title is Engineered EVs as mRNA Cancer Vaccine Delivery Platform Conferring Immunomodulation in HCC. Please welcome.
Okay, thank you. And my apology that I have changed my title a little bit ago. You can see the title is here. Engineered EV as Human OX40L mRNA Delivery Platform Conferring Enhanced in situ anti-cancer efficacy of TILs in HCC. This HCC is hepatocellular cancer. So here is today's topic. Okay, nowadays, the single-cell sequencing reveals the distinctive functional composition of T cells in tumor microenvironment, and several studies have proved that the enrichment of T cells in the HCC tumor, especially the Tregs and CD8 positive T cells. And we found that the mentioned infiltrated CD8 positive T cells are mostly exhausted and have innate-like no cytotoxic and no clonal expansion phenotype. So we wonder if there is any way to modify them, to reactivate them, to have some use for the therapeutic for the HCC therapies. Apart from the well-known checkpoints such as PD1 and PDL1, we found another positive immunocheckpoint, that is OX40 and OX40 ligand. This interaction could activate the immune system, and here the OX40L, that is OX40 ligand, has been identified as a co-stimulator for the T cell activation. And our previous study has proved the overexpression of the OX40 ligand in HCC cells could promote the activation of OKT3 primed T cells. Then nowadays, the mRNA-based therapy emerged, and we know that extracellular vehicles could provide a suitable platform for the mRNA delivery. So the study has been designed to use the engineered extracellular vehicles to deliver our target mRNA encoding human OX40L specifically to the HCC cells via the HN3-GPC3 interaction. HN3 is the antibody for the GPC3, and GPC3 is overexpressed on the surface of HCC cells. So by this way, this HN3 and GPC3 interaction could guide our engineered EVs to the HCC cells specifically. Then two sets of the RNA binding protein and relevant element has been selected. That is LA7E box CD system and TAR and TAT system to deliver the interest mRNA. And ACGMP mRNA has been used as a control in the study. So we have generated our donor cell lines. You can see here, for example, the donor cells. Here the donor cells we call it AT. That is, it has expressed the HN3 antibody and used with TAR and TAT system to deliver ACGMP mRNA. And here, HB cells refers to the cell expressed HN3 antibody and used LA7E box CD system to deliver human OX40L mRNA. So once the donor cells are ready, we have collected EVs and purified them and do the normal characterization. Here is the TEM image and NTA analysis that the EVs are nanoveticles with biolayered that round veticles. The diameter is around 120 to 180 nanometers. And the Western blot has analyzed the well-known that EV markers. Here we need to pay attention is in the HT donor cells and the HB donor cells. Please see here. That is human OX40L protein. We can only take the band of human OX40L protein in the donor cells, but not in their derived EVs. Also, we have extracted RNA from the EVs. See the figure G here. In our control EVs, there is no human OX40L mRNA detect. And compared to the HT-derived EVs, our HB EVs carried more copy number of the human OX40L mRNA. Then to check the specific targeting ability of our HN3 EVs, we have co-cultured the AO2 cells and the HH7 cells, one of the HCC cancer cell lines. We can see here, AO2 cells didn't express any human GPC3, and the HH7 cells express the human GPC3 protein, and we have modified to express the mCherry fluorescent tag. Then the EVs has been pre-stained with the green fluorescence and then add to the co-cultured model. Then after the incubation of 2 hours, the cells has been collected and has been checked. This is the FACS analyzed. We can see compared to the red bars, that is our controlled EVs. The HN3 paired EVs are more gathered in the HCC cells. Also, we can see in the conformal mixture, the first layer, that is our controlled EVs, and it didn't show any bias to any type of the cells. The bottom layer, that is our HN3 expressed EVs, they are more gathered in that red cell, that is our HCC cancer cells. Also here, this is the in vivo picture that shows the same thing. Our HN3 modified EVs are more enriched in the tumor site. Then to analyze whether our EVs can deliver the human OX40L mRNA to the target cell functionally. We have incubated the EVs with the cells, and then at different time points, we have collected the cells for the analysis of the human OX40L expression, both in mRNA level and the protein level. See the figure A, C, and E, that is after treatment of the HB-derived EVs in the target cell or in the recipient cell, the mRNA level and the protein level of human OX40L got increased in a time-dependent manner. But that B, D, and F, that is after treatment of HT cell-derived EVs, the results are altered. That may be because of the insufficient encapsulation of the human OX40L mRNA. Also, we have to do some immunofluorescence to confirm the expression of the human OX40L. So after the treatment of the HBEVs, our recipient cancer cells got the expression of human OX40L on their surface. So the HBEV was selected for the later research and hence donated as EV-OX40L. Then we have co-cultured our EV-OX40L with the OKT3 primed T cells and compared to the control EV, that is ABEV, our EV-OX40L has performed well in the activation of T cells. You can see the spheres here, that means the T cells has been well activated. Also, the activation marker CD25 has been checked. Here is the FACS result, and compared to the control ABEVs, the CD25 percentage is around 25 something. And after treatment of our engineered EVs, the activation has reached out 60%. Also, we have introduced the whole system to the HH7 cancer cells to see whether our engineered EVs has any cancer-killing effect. So we have mixed the T cells and the HH7 cells at different ratios, and compared to the untreated group, we can see the second bar. That is our EV-OX40L treatment. That treatment has greatly increased the cell death number. Also, we have added the OX40L blocker to block the OX40 and the OX40L interaction. And as expected, this kind of blockage reversed our engineered EV-induced cancer cell killing. Later, the in vivo study has been performed too. Here is the schema for the in vivo study. At the beginning, we have given the mice, the NCG mice, PBMCs, and we have checked the CD3 and the CD45 population every 2 to 3 days. When this population is stable, the HH7 cancer cells has been harvested and given to the mice to grow tumor. And when tumor is ready, we have given the EV treatment. The tumor has been checked or monitored every 3 to 4 days. This is our first round animal study result. You can see from the left to the right, here is the MOC group, here is the NC group, and here is our EV-OX40L group. So from the size itself, we can say our engineered EVs performed very well to control the tumor growth. Also, the tails has been extracted from the tumor, and we have checked some phenotype or the immunocell population. Here is the result. Compared to the negative control, our engineered EVs greatly increased the CD8 positive T cell population. Also, it has increased the CD4 positive T cell populations too. Especially in the CD4 positive T cell population, we have checked interferon gamma and CD25 and FOXP3. Compared to the negative control, the interferon gamma population got increased too. That means the Th1 population got increased. And CD25 and the FOXP3 population got decreased. That is the Treg cells. So in conclusion, that is our engineered EVs, EV-OX40L, has increased the Th1 population, which could further promote that CD8 positive cytotoxic T cell and enhance that cancer killing efficacy. Also, the Treg cells got reduced. So the whole that is, I can see here, so the whole immuno microenvironment has been changed in a positive way, and more analysis should be drawn, and we are doing the things. So, okay, thank you for your attention.
Excellent presentation. presentation, and it's time for questions, please.
Thank you so much for your diligent work. I was wondering about, since you're activating the T cells and decreasing the Tregs in your tumor microenvironment, but since there could be a bystander activation and like a kind of an autoimmune response, have you checked if you've seen any autoimmune responses with the immune checkpoint or the things that we now see, the immune-related adverse events?
Actually, we have checked some Brazine and interferon gamma and leukemia 2 and some other inflammatory factors, but with ELASA, and the result I didn't show here.
So, like, since they're increased, do you see any autoimmune adverse event in other tissues or surrounding tissues?
Just in tumor.
Okay, thank you.
Thank you.
Yeah, very nice talk. So, may I ask that if the donor cell, the EVs are 293, but not the liver cell or tumor cell?
Just 293.
What is the reason that you use the 293 cell therapy EV for the HCC treatment?
Actually, we have done some work before, and we have compared the 293 EVs and the tumor-derived EVs. Tumor-derived EVs has a tropsen to the tumor itself, but it could bring some molecules that will increase the tumor progression. That work I didn't show here, but we have published.
Okay. And the next question is for the different ratio, the co-culture T cells and the tumor cells. Yeah. The tumor cells are treated with the EV from 293. Okay. So did you find any the ratio affect the response of the T cell to the tumor killing rate of the T cell? The ratio of the tumor cells of T cells, the co-culture.
Tumor cells with T cells. Oh, you mean the in vitro study?
Yeah, in vitro study.
Here? You mean here?
Yeah.
Oh, actually we didn't do this kind of set too, so I didn't do that.
Okay, thank you. I have one question. Yeah, because OX40 and OX40 ligand are well-known strong co-stimulator in immune response. So I'm wondering the side effect after your engineered EVs delivered to the bodies. Maybe some of the EVs can circulate to the whole system. Side effect because this OX40 and OX40 ligand is the co-stimulate, just like the CD80 and the CD86. Yeah.
Yes, that is the problem with phase 2, and we are thinking to design that switch on and switch off system to make like once the EVs meet the tumor cells, then it will switch on, and then this OX40 ligand should be work in that way. That is our next level work.
Yeah, and my another question is, even you deliver the mRNA and to you use the EV to deliver mRNA. So my question is, how about the long-term effects for these EVs after you transplanted the EV to the tumor? Long-term effect.
Actually, that's here. That is the day 27, and just one injection.
Yeah, because the mRNA if the mRNA was delivered to some tissues and some protein will be produced based on this mRNA. So it should have the long effects after the administration.
Yeah, but we think that mRNA could be more safer as we have the problem. The EVs may scatter to some other where, so other cells may express the things and may cause some inflammatory reactions. So we use the mRNA to avoid that kind of problem. But I think the next level, if we have finished that switch on and switch off design, it will be better.
Okay, thank you. And any questions? Okay, that's again thanks to Dr. Chong He.
Our next speaker for today is Dr. Rosmarin van den Broek, who will be speaking to us about extracellular vesicles and their role in Alzheimer's pathology in mice. Thank you.
So, good morning. So it's a pleasure to present some of our data here today related to MSC-derived EVs and their treatment for Alzheimer's disease in mice. So, as you probably all know, MSC EVs are known to have a lot of potential therapeutically, and they are involved in tissue regeneration, and they're also known to have an impact on the immune response. So they have this inherent anti-inflammatory capacity. So my lab is interested in Alzheimer's disease. And as you probably know, the typical hallmarks of Alzheimer's disease are the A-beta plaques and the neurofibrillar tangles. But next to that also there is neuronal dysfunction. And we are very much interested in the aspect of neuroinflammation. And it became clear the last years that neuroinflammation is probably even an early event in Alzheimer's disease pathology. So this made us wonder whether these MSC-EVs could be a potential treatment to treat Alzheimer's disease early on because of these anti-inflammatory properties. So MSC-EVs, they might have therapeutic potential, but there are also some problems at this moment. So there's a lot of variability described in their therapeutic potency. So this is mainly due to the heterogeneity of the parental MSCs, and this results in a low consistent production of MSC products. The lab of Bernd Giebel invested a lot of time in generating or in trying to solve this problem, and they generated immortalized MSCs and then globally expanded them to really have a robust EV production system. So this has the advantage that there will be less variation in the therapeutic potency of the MSC EVs. And they recently published that these MSC EVs maintain their therapeutic properties, and this was tested in a mouse model of hypoxia-induced neuroinflammation. So in our study, we started from a specific mouse model of Alzheimer's disease, and I want to explain it a bit more in detail. So we in our lab make use of this, of these knock-in mice, so these APP and LGF mice. It's a bit different of a lot of the other Alzheimer's mouse models where they use overexpression of the mutant APP. So in our case, it's endogenous expression, so there are less artifacts associated with this mouse model. In addition, a problem is that mice kept in the lab are often, let's say, too clean. So it's not realistic. It's not like in the human situation where we are continuously encountering infections. So several years ago, we started introducing a kind of low-grade inflammation in these mice by injecting LPS intraperitoneally. So it's not a very high inflammation, it's low-grade. We do it 2 times. And we've published that after 2 weeks, you see aggravation of the A-beta plaque deposition. So adding this low-grade inflammation induces more plaque formation, and if you quantify that, it's you see this increase in plaque area. And also, if you look at the microglia, these innate immune cells in the brain, this is how they look in more homeostatic conditions. But if they are triggered, if they are more activated, they look quite different, and you can quantify this. So we know that in this mouse model, there's more A-beta plaque deposition, and the microglia are more activated. And we really believe that this model is more representative for the human situation. So this is the mouse model that we used here. So what we wanted to do is use these MSC EVs as potential treatment for Alzheimer's disease. So EVs were isolated according to the MISEF guidelines. And it's also important in this case to use the proper control EVs. So the MSCs are grown in the presence of platelet lysate. So as a control, we used human platelet-derived EVs in our treatment. So we had our mouse model, the APP and LGF mice with low-grade inflammation, and we intranasally delivered the EVs every other day during these 3 weeks. And then we performed the analysis. So first of all, we looked at the cognitive function of these mice. So this is one test, the Morris Water Maze test. And here you see in blue the mice treated with human platelet-derived EVs, so the controls, and then in red the MSC EVs. And so you test the mice all the time, so you look at the distance they swim to reach the target, so the hidden platform. And you see that the MSC EV-treated mice showed better spatial learning. You also do these probe trials at different time points, and here I show, for example, the probe trial 2. And on the left in blue you see the human platelet-derived EV-treated mice. And if you look at the MSC-derived EV-treated mice, you see that they have a better reference memory. So the time that they spend in the target quadrant is higher compared to the other quadrants. So the MSC EV treatment has a positive impact on the cognitive decline. So what about the A-beta deposition? As I mentioned, this is one of the hallmarks, of course. So here you see 6E10 staining, so the A-beta plaque staining in green. And this, if you quantify that, the treatment resulted in a decrease in the amount of plaques in the mouse brains. If you had a closer look to the plaques, in red you see now the microglia. As you already see in these images, and also the quantification confirms it, and that was especially the case in the large plaques, you see more microglia associated with the plaques. So clearly the microglia that are associated with the plaques are trying to move the A-beta or to degrade the A-beta. So the treatment has also a positive effect here. But we also looked closer to the microglia and we tried to quantify the amount. So despite the fact that there were more associated with the large plaques, in total in the brain there were less microglia in the brain. And related to their activation status overall in the brain, so this is again this morphology that I mentioned before that you can obtain via 3D rendering, and you can quantify that, so you can quantify the branch length, amount of branch points, etc. And here you see that several of these markers are increased, showing that there is a decreased microglial activity. We also looked, I don't show it, but we also looked at astrocytes, and also there you have the same effect, so decreased activity in response to the MSC EV treatment. What about this neuronal network? Is this also affected? So we looked, for example, at Neu-N staining, so you can quantify the total amount, and this revealed that the density of the neurons is increased, so the MSC EV treatment has also here this positive effect. Neural network can also be quantified by this co-staining, so you stain the presynaptic and the prosynaptic elements, and then you quantify the amount of double positive dots. So here you see the PSD95 and CYP1 staining, and if you quantify that in different regions, you see that this is increased. So there are more synaptic puncta, showing that the neuronal network is better in response to this MSC EV treatment. So, as I said, we are very much interested in inflammation, and it's known that during Alzheimer's disease there is an influx of immune cells into the brain. And we also quantified this in the MSC EV treated mice. And this revealed in total a decrease in amount of immune cells. But we also had a closer look to which cells were then affected. And if we look to the innate immune cells, both the amount of infiltrating macrophages and neutrophils is decreased in the MSC EV treated mice, while the amount of DCs and monocytes was not affected. And also in the adaptive immune cells, there was a difference. So here you see B cells, NK cells, T cells, and then we also looked more closely to the CD8 positive and CD4 positive T cells. And only in case of the CD8 positive T cells, we saw that there was a decrease. So MSC EV treatment reduced the amount of infiltrating CD8 positive T cells in the brain. So we're very much interested in where the MSC EVs are traveling, because okay, they have this positive effect, but how is this possible? and where do they go into the brain. The system was already explained during several talks, so just shortly, so what we do, we have our MSC EVs, and we post-load them with Cre protein. So we do this with the saponin method. It's a method that works, it's not super efficient, and it might also affect, of course, a bit the EV surface, but that's the best way that we could load the MSC EVs in this case. We again purify it by SEC, and here you see indeed that in the EV fractions you really have the Cre, so it's loaded in the EVs. We always test it up front, whether it's loaded and whether it's working, and this is a control condition, and you see an increase in amount of red cells in case of incubation with Cre-loaded EVs. So we use these Cre-loaded EVs and then inject them in the TDT reporter mice. These mice have the system where you have a flux stop in front of the TDT gene. So only when the Cre is present in the cell, delivered in the cells will turn red. So that's a powerful approach to really visualize where the cargo is delivered into the cells. So we use the system, so we use these TDT reporter mice. We treated them with either MSC EVs or platelet EVs loaded with this Cre. We did that on 5 subsequent days and then did analysis at day 7. So you can visualize that, so you can visualize the amount of positive cells. We did intranasal delivery, so we of course looked at the olfactory bulb, and there you already see an increase in red cells. Also in the prefrontal cortex, we saw an increase in amount of red cells in case of MSC EVs compared to platelet-derived EVs, and also in the hippocampus. In the cerebellum, we did not see an increase in Cre-positive or TDT-positive cells. You can also quantify this, not by just looking at the images, but really by doing flow, and that really allows you to analyze a large region of the brain. So we quantified the amount of RFP-positive cells, again comparing the platelet EVs with the MSC EV treated mice. And you see in all cases that there are more red-positive cells in case of MSC EVs. So clearly the EVs are able to deliver their cargo to the brain. So this brings me to my conclusions. So we've shown that intranasal administration of these MSC EVs to the APP and LGF mice improves their cognitive functioning, decreases the amyloid beta pathology, decreases glial cell activation, and improves neuronal dysfunction. So related to the mechanism, we've seen that this is associated with a decrease in immune cell infiltration, mainly macrophages, neutrophils, and CD8 positive T cells. And we've shown that they are really able to deliver their cargo throughout the brain. Of course, there are still a lot of questions, or a lot of open questions. So currently we've done intranasal administration because that's of course a way to get into the brain. However, whether that would also be possible via intravenous delivery is less clear. We've done that in the past in another mouse model, in a mouse model of NPC, and there we had positive effects on the brain, but at that time we did not check whether these EVs were able to deliver their cargo to the brain. And of course the question is always, what are the key molecules in the MSC EVs that have this therapeutic effects? And that's a difficult question to answer because probably it's more a combination of molecules rather than one molecule that has the impact. And then the fact that of this cargo delivery, is this really essential? Well, we've shown that it's happening. Maybe it's not even needed. Maybe it's enough that the EV can reach a cell and just interact with the cell, interact with the receptor, induce some signaling. Maybe that is even enough for the therapeutic effect. And with that, of course, I have to end by thanking all the people, so the Barriers and Inflammation Lab, and especially Lien van Hoeke, postdoc in the lab who did most of the work, and this was of course a close collaboration with the lab of Bernd Giebel, and I will be happy to take any questions.
Thank you, Doctor, for that great talk. We'll now open the floor for any questions.
so the clone that was selected was the one that had in vitro the best anti-inflammatory properties. Indeed, that was the reason that we used that clone. So it's the, let's say, optimal clone which had on inflammatory cells the best anti-inflammatory capacity.
And the other question I have is, you showed the intranasal one, probably that's why you checked like only the CNS part, but have you checked other tissues if the Cree is giving any signal?
No, we didn't. I would assume that there's not much peripheral delivery, but I agree, this is something that we should check. We didn't.
Yeah, because one of the reasons I was wondering is that since they're kind of an immunosuppressive environment and other infections would be like worse. But thank you, great work.
Thank you for this nice presentation and interesting data. So with regard to intranasal application, how would you deal with the fact that if you have a mouse, you have a surface epithelium area, which is the ratio between the surface area of mouse nose to body volume is a completely different story than in human beings, and how to translate?
Yeah, you cannot translate it, that's indeed the problem. So we really wanted to do this approach to show whether they have this activity. In my lab, we're really interested in improving or increasing the brain targeting potential of MSC, of EVs in general. So for example, we've been investing in generating ligands that really induce brain targeting. So the idea would be to modify the EVs to have brain targeting potential even upon IV administration rather than intranasal. Because I agree, I mean, you cannot translate the mouse data and the distribution throughout the brain with human brain.
Ever considered intrathecal application?
Yes, we do that too. We have these mini osmotic pumps that we use, and then you have direct administration. And we know we've done some experiments with other EVs, not with these MSC EVs. And then we have a very efficient delivery throughout the brain, or in some specific cases at least. So it is possible, but to have it therapeutically applicable, I would prefer an IV administration rather than an intrathecal administration. If you have to do that repetitively, it would be a problem.
We disagree. I prefer intrathecal.
Yeah, but it's invasive. I mean, I agree, but
as an MD, that's okay. You can do this if it's against such a disease.
yes, okay, that's true.
Thank you. It was really interesting to see the positive effects on things like the amyloid pathology. I was wondering though, I don't know how much of a chance you've had to look at the individual pathways that might be affected, no? But do you think that this is maybe specifically related to the production of amyloid, or is it more of a general effect? I know that MSC-derived exosomes have been shown, I think, to deliver things like catalase, which are obviously more generic stress.
So we haven't looked into specific pathways that are altered. It's not an easy setup, of course, to look at these aspects. What we do think is that the microglia are somehow still, let's say, in a better shape to phagocytize the A-beta, so that they can cope better with the A-beta that is accumulating. That's really where we see, I mean, despite the fact that we see less microglia and less activated microglia throughout the brain, they are more associated with A-beta plaques. So they are better doing their job compared to in the non-MSC EV treated conditions. So maybe that's the impact that the MSC EVs have directly on the microglia or via the immune cell infiltration that is less, I mean, generating a better environment for the microglia to function.
Yeah, okay, thank you very much.
Awesome, that's all the questions. Can you please join me in thanking our wonderful speaker for sharing?